Initially, the G protein link receptor, one of many receptors in the cell membrane, bind to a ligand, which is a signaling molecule. These receptors are very specific, meaning that the shape of the binding area of the receptor has to be very specific. Once it binds to the receptor, it becomes activated, and the shape of the receptor changes. This commences the transduction pathway. An inactive G protein binds to the area of the receptor in the cytoplasm that has changed shape. Once it binds to the receptor, the GDP on the G protein is catalyzed into a GTP, thus activating the G protein. Then, it makes its way to an inactive enzyme, adenylyl cyclase, and activates it by binding to the enzyme. The adenylyl cyclase then converts the ATP to CAMP, which happens to be the secondary messenger in this case. CAMP then binds to a protein kinase, activating it. This molecule then phosphorylates other proteins to initiate the response part of the pathway. In this case, the response is the signaling of the smooth ER to release the calcium ions. Ligands first bind to the tyrosine kinase receptors embedded in the plasma membrane. This activates the tyrosine kinase receptors, resulting in them coming together to form a dimer. This begins the transduction pathway. Then, ATP arrives and phosphorylates the receptors. Then, inactive relay proteins come to relay proteins and bind to the receptors. This activates the relay proteins, so the active relay proteins go to the smooth ER. Finally, the response. Response in this case is the release of calcium ions into the cell. Parkinson's disease is a result of error in synaptic signaling. The neurotoxins, which are neurons affected by the environmental toxins and reduced dopamine, bind with the receptors embedded in the plasma membrane, whereas normal ligands should bond with the receptor. This results in an excessive amount of ROS, which stands for reactive oxygen species. These molecules make their way to the nucleus, which then cause some specific genetic mutations. Without excess ROS, an enzyme would be activated, resulting in normal function of the cell. The DNA replicates to make mRNA, which then makes its way to the ribosomes on the rough ER. The ribosomes translate the mRNA into proteins and carry on the mutations while make the protein. Because of the genetic mutation, this results in a protein called alpha-synuclein, which is essentially a misfolded protein. Once released into the cell, they transform into Lewy bodies, which are abnormal clumps of a protein or proteins, and the proteins cause Parkinson's disease.
Disease is very slow but progressive. Initially, it would result in signs of weakness, stiff muscles, and difficulty walking. Tremors also may or may not be existent. These symptoms eventually and gradually worsen. Other symptoms may include slowness of voluntary movements, decreased facial expressions, decreased rate of blinking, spontaneous speech, and abnormal walking with swinging arms and stoop posture, unbalance, difficulty standing and sitting, and swallowing problems. As of now, there are no treatments for the disease. However, scientists are researching methods of slowing the symptoms down. Developing treatments include a pharmaceutical approach, surgical approaches, gene therapy, and cell implantation.